Thanks, Marcel, and thanks to everybody for sticking around and for organising. So I'm going to give a high-level overview to finish up today on Nextstrain and how we've been tracking the coronavirus pandemic over the last couple of months. And I think it bookends quite nicely with the way we started this. Started today with Eddie's talk about finding the virus and discovering the original zoonoses. So three sort of parts to the talk briefly. What is Nextstrain? What were we up to before this pandemic? How did that position us quite well, I think, to be able to respond, and hopefully how we have been useful to the wider community, and then finish with a few slides about the direction we are trying to push things or help people along with over the coming months. And before I get to that, it's really important to say that all of the work that I'm presenting here is a result of a collective effort from many wonderful people involved in Nextstrain behind the scenes, and that all of the data, all of the sequence data that we are using has been contributed by scientists all over the world, including quite a few who have been talking today. I think there's like 1,000 or more genomes now from Australia, so Australia is really well represented in the global data set, and it's all being shared through, or primarily being shared through GISAID, which is how we're getting our hands on the data. So what is Nextstrain? So I've been a postdoc working with Trevor Bedford for 3 or 4 years now on Nextstrain, and the idea has been that we would like to do genomic epidemiology of emerging and ongoing outbreaks and epidemics. We want to do it in a rapid fashion, and we want to be able to update that as new data is available and share those results as widely as possible. And I think it was summed up quite nicely in a message from Ollie Pybus quite recently, where he said that Nextstrain is built on decades of research by the wider phylogenetics community, and is a good story of basic theory being transformed into applied science. So how does this work in practice? So we have a number of different sort of databases or sources of data that have been publicly shared through GISAID, through other platforms as well, some of which might be shared through emails in a sort of semi-public, semi-private fashion. We grab that data, we run bioinformatics tooling, which we call Augur, the internals of which a lot of you will be familiar with. They're well-established tools such as MAFT and IQtree. And then we use Auspice, which is our interactive visualization software for our own purposes, to interrogate the data, to explore what the data is telling us. But because it's web-based, we also use it to disseminate the data through nextstrain.org. And so we have research interests for data sets mainly centered around RNA viruses, but not exclusively. And we keep these data updated. So things like seasonal influenza, avian influenza, West Nile, Zika, mumps, and quite a few more. And so as new data is available, be that every day, be that every week, be that every month, we can rerun the analysis and share that data through nextstrain.org. But it's not just a sort of centralized project just for us and our research interests. We've got a lot of interest in exploring making this a tool for the community and getting the community to actually drive it more than us. So we've got a number of data sets that are being completely maintained by members of the community, to the extent that they can update the data and share it through nextstrain.org without us even knowing. So there's no day-to-day interaction from us with these community data sets. And I want to just point out a really fruitful collaboration we've had over the last couple of years with the INRB in Kinshasa in the Democratic Republic of the Congo, who, with the unfortunately still ongoing Ebola outbreak, they are not only sequencing Ebola genomes in country, they are running all of the bioinformatics analysis using Nextstrain in country and sharing that data with the wider community through nextstrain.org, all without any day-to-day involvement from us or foreigners. And I think it's no coincidence that the DRC has been one of the best countries in Africa in terms of being able to turn around that capacity and sequence and analyze coronavirus. So I think that positioned us reasonably well to respond quite quickly to what was then called nCoV. 1 or maybe 2 days after the original sequence was made publicly available, Trevor had spun up this build of all of the publicly available beta coronavirus genomes, and really clearly seeing how the coronavirus, or the novel coronavirus, fitted into the background variation. And this figure is broadly similar to the one that Eddie showed at the very start of this talk. And so ever since then, we have been updating the Nextstrain build, or the Nextstrain view, into SARS-CoV-2. And the high-level overview of how that actually happens in practice is that, as I said, people from around the world are sharing their data publicly with GISAID. Every 15 minutes, we programmatically check if there's any new sequences. If there are, we rerun the analysis using Augur. So Augur tries to toe the line between speed, because we want to do these things rapidly, but also maintaining scientific validity and confidence in the data. So for a 3,000 genome set of coronavirus, we would be able to run that in a couple of hours just on our laptop. And then the results are pushed up to nextstrain.org. And so in the early days, we were sort of updating the website almost a dozen times a day as new genomes were coming in. That slowed down a little bit. Now we're sort of trying to do it every day, or maybe twice a day. So if you go to the website, this is a screenshot that I took yesterday. You can see a presentation or the visualization of the current worldwide spread of COVID-19. It's about 4,000 or 5,000 genomes, I believe. And you can see the phylogenetic tree on the left here, where the horizontal axis represents time of sampling, and each little dot is a coronavirus genome, complete genome, colored by the place around the world, or the region in the world in which it was collected. And at the bottom there, you can see a bar chart showing the diversity of mutations as we observe it in the data across the sort of 30 kb genome. And the idea is that this is completely interactive. This isn't just a static image that's updated every day. It is a living, breathing data set. And so you can zoom into different parts of the tree. You can restrict the data based on different attributes in the metadata. You can change the coloring to represent other fields in the metadata. You can scrub through time to see how it's spread around the world across time. And I'll just do one of these for you now, which I thought was quite interesting, which we haven't actually touched on in today's talk, which is to look at one mutation in particular. And so there's been a lot of stuff in the media about this mutation in the spike protein, which has all sorts of effects if you believe the headlines. But what this allows you to do is to find that in the diversity panel, click on it, and now you're coloring the data by the mutations which we observe at that position. So this is amino acid 614 in the spike protein. And you can see that in the bottom here, we're seeing some comparisons of PCR cycles needed to amplify for the different variants. And there does seem to be a slight effect there. But we can now explore that data and see how do these variants distribute around the world, and how do they distribute across the tree. And so we can start exploring the data in more depth and making our own hypotheses. And if we mouse over the branch where it switches from amino acid D to G, we can see that that's associated with a transmission event from China to Europe, and one of the main events that seeded the quite large European outbreak. So we can immediately see that what we're potentially seeing here in terms of the change in frequency could well be attributed to founder effects rather than some selected variant growing faster. So that's interesting, and I think it's been really valuable to the wider scientific community, but it does require a certain ability to read and interrogate phylogenetic trees. And so one of the things we've been focusing on is writing these interactive narratives each week to sort of explain how it's moving around the world and what we're seeing in the data. So we write these in each week in English, and we've been really impressed that the sort of through Twitter and GitHub, this community's come together from around the world and each week volunteers translate these reports. I think now they're in about 20 languages or so. That's being done just by the community, which is cool to see. So what do these interactive situation reports actually look like? So this is a one from last month. You can find all of these on nextstrain.org if you're interested. And they're basically us telling us a story, walking through the data as we see it. So they start off looking somewhat akin to a blog post or a scientific paper. But on the left, as you see it, you've got text, and on the right, you've got the visualization of the data. And as we change paragraphs of text, the view into the data changes as you scroll through the different paragraphs. So here in this narrative, we were focusing on the worldwide spread. So looking at early samples, spreading within Asia, scrubbing forward in time a little bit and talking about how Asia seeded introductions into other regions, and then having slides which focused on more bespoke questions. So where did regions such as South America, Africa, and Oceania get their samples from? Where were the majority of introductions from? And so in general, we've been trying to communicate things, a lot of which have been talked about today. These ideas that coronavirus was disseminated widely before travel restrictions came into effect. It typically was spreading before it was even noticed. We're trying to do lots of work to explain what mutations mean, that they're not actually terrible, but also that with the mutation rate or substitution rate we're seeing, it's too low to resolve direct person-to-person transmissions quite often. And a lot of energy and time invested into sort of trying to debunk some of the conspiracy theories or just the sort of ideas out there that the data don't support. And I think we've been had a reasonably good platform to be able to debunk some of these by showing the data next to the claim that we're making and seeing how it doesn't add up. And so we're getting more and more sequences, which is brilliant. But where do we go? So this main figure here is showing all of the sequences that have been submitted to GISAID so far. And so the red dots over time you can see climbing up the graph are the sequences, when the sequences were made available in the database. And then if you trace back horizontally in each row, the blue dot is when that sample was, or when that sequence was sampled. And so you can see that there's some lag here. We're sort of seeing a lag of about 2 to 6 weeks between sample collection date and the sequence being made available publicly. But I think it's fair to say that this is the closest to real time that we've experienced so far, especially on this kind of scale. And if you look at the vertical axis and the number of genomes, and even just look at the shape of the graph, the graph is heading up really quickly. The number of genomes, we're up to about 25,000 genomes, which is fantastic. But 25,000 genomes has its own problems, both for analysis reasons, computationally, but also for visualization and how we actually understand that many sequences. So up until about mid-April, we were making daily or more than once a day updates to nextstrain.org. We were tracking all of the data that was being uploaded to GISAID, and it was going really well. But the number of genomes was just growing too fast. And so since early April, what we've been doing is subsampling the data. So still presenting a global analysis that's up to date, but is heavily subsampled. And also producing builds for each of the different regions around the world, subsampled to focus in on genomes from those regions, but also provide some sort of worldwide background context as well, which is really important. And what we want to do over the coming months, and what we are doing already, is to try and facilitate state, regional, county research labs to run their own analyses, which will help them ask and answer their own sort of questions that are very relevant for that part of the world. You know, there's a lot, I think a lot of talks today are focused on how useful that can be, especially Torsten saw just before. And we, I think, are able to provide advice and tools and just help people out with this. and also allow, it's all open source. All of the tools are completely free, available to you. So you can do it privately, but we also have nextstrain.org available, and the reach that that provides. We're trying to encourage people to share their data through that platform, both in terms of raw data and the phylogenetics, but also in terms of writing these reports to disseminate their understanding. And I'll just finish with two pictures of such things. So the left-hand side is a group in Spain who have called themselves Nextstrain, who are using Nextstrain to spread their data and understand some questions relevant on their geographic scale. And then on the right, we've got an example from Jope's talk earlier, which is using Nextstrain, but just with, we've got functionality where you can have private groups. So you need authentication to be able to see the data, which is currently the case for the New Zealand sequences. And with that, I'll finish. Thank you very much for sticking around, and just emphasize that this was a really collective effort, and say thank you to everybody who's contributed sequences to the global collection. Cheers.